Hello, everyone. My name is it showing correct? Okay. My name is Qian Liu, and I come from the Bioconductor core team. It is located in Buffalo, the city of Buffalo in the United States, if anyone interested. And thank you, BOSC, for having me. And today I'll talk about the lazy representation and comprehension of very large genomic data resources and our Bioconductor. So Bioconductor is an open source, open development platform for bioinformatics tools and statistical genomic tools. And it is based on our programming language. That's why I'm a street R user, and I hear everything. I hear anything similar to our SR, so sorry for not paying attention to your talk. And I was doing my last minute rehearsal. First, I'll introduce the Warren experiment. It is a data container for your assay data. Basically, it extends the summarized experiment, which is familiar to many bioinformaticians. And it extends summarized experiment, which means that it inherits all the methods that are already defined on the summarized experiment for free. And there are three major components for the Warren experiment and summarized experiment. The first is the assay data, like it is in the middle, your major experimental data. It could be the genotype data from GWAS, it could be the dosage for variants from your DNA sequencing, it could be the count data from your RNA-seq or single-cell RNA-seq. The other two components, for your assay data, the rows are for your features, which include the genes, probes, or variants, and the columns are the samples. So the other two components of summarized experiment are the two annotation files about your features, which could be called as the row data, and your column data for an annotation file for your samples. And there's a metadata on this corner, which means it is a list. It's an R list, which means that you could put anything related to your assay or experiments here. These are the common things between summarized experiment and Warren experiment. So I talk a little bit about the difference. So when you use summarized experiment, basically when you load your data into summarized experiment and do your analysis in R, you load everything into memory. I mean, mostly. There are some exceptions. You load everything into memory, including your assay data, including all the annotation data. But when the data gets really large, and it's very difficult to fit into the R memory limit, so that's why we design the Warren experiment. The major difference would be that it is a container for the lazy data structures. So it represents the assay data as a delayed array and any extensions. The backend data could be a SQL database data. It could be a GDS file format. It could be a very big VCF file. It could be H5 file that there's an interface called HDF5 array. And the annotation data for the features and samples are represented using the delayed data frame. It is a lazy data structure for containing those different data types. With, for example, for the samples, you can have their gender information, their age, their cancer stage, the disease status, their family history, anything. They are in different data types, and they could be lazily represented inside the delayed data frame. So with everything I have said, you don't really need to understand, because we have very convenient conversion functions. Okay, we have a function you can directly convert your VCF file into Warren experiment, and you don't need to anything about. Our aim is we don't want users to feel any difference when you deal with the data in memory or out of memory. I'll talk really quick about the SQL data frame. It is another data structure that extends it's the data frame-like interface. So many core Bioconductor packages implemented the data frame, like variant annotation, genomic alignments, genomic ranges, summarized experiments. So it could be very useful when you use, even if you don't notice, it could be very useful. So the idea is similar. We want the lazy representation of SQL database tables. For example, we have a SQLite data from the ORG-DB for Homo sapiens, and table is gene info. So when we represent the data using SQL data frame, so the data looks like this. So it's very neat. You have the a show of the first several rows and last several rows. And we load another transcript DB for Homo sapiens. And you can use the function inner join to join these two SQL data frame and get all the information you want. So everything is lazy. The data are stayed on disk and unchanged. So I want to thank Martin Morgan. He's the director of Bioconductor, and Irvi, he's the developer of delayed array, and many other fundamental Bioconductor packages. You can ask me any questions through my email, and thank you all for your attention.
Okay, thank you everyone for your good and on-time talks. So I have a question for Sapporo. Is there a particular project reused to provide the WES implementation, or was that built by the Sapporo team?
The implementation of the WES is implemented by ourselves. And I think the Sapporo service is one of the first implementations of the workload execution services. But we made a slight modification to the WES API because we don't want users to add their own workload. So we just delete that part of the API. So it's not a perfect implementation of the WES, but you can still use that.
Thank you. Bioconductor. Can I use variant experiment for managing single cell data?
Hopefully in a couple months. So you can use a single cell experiment. So this is another data structure that directly extends the summarize experiment. The difference is that the single cell experiment could represent your assay data in HDF5 file. So the assay data is lazy, although the annotation data are not lazy. So you could manage your data, but the annotation data are loaded into memory. If you have really big annotation data, maybe you need to think about the variant experiment. And I want to support in the future for the count data with with the HDF backends in variant experiment.
Thank you. And one for ROG. Is it possible to use it to create a CWL file without explicitly using the graphical user interface?
No. So the main purpose is to generate a workflow from the selection of tools. So that wouldn't be the point of ROG. Thank you.
Okay, thank you everyone for the questions. So just as a reminder, we're about to move on to the next set of speakers. If you want to ask a question, you can go to sly.do and then enter the code BOSC 2019 to ask questions. So we have two more speakers up, and the next one is Moni. So